Hello everyone, welcome back to our 7th video lecture here for Unit 4, Photosynthesis. In this lecture we're going to discuss the second step of photosynthesis, which is referred to as the Calvin cycle. Alright, so over here we have Dr. Melvin Calvin, who discovered the Calvin cycle and won the Nobel Prize for it. So our goal during this process is going to be to make high energy sugars such as glucose. Alright, when I say high energy sugars, we're going to see later on in cellular respiration in the mitochondria that we can break these sugars down to produce an exorbitant amount of ATP. But we're going to make these sugars by removing CO2 from the air and use energy from the light reactions that we produce, both ATP and NADPH, to make sugars. Alright, so what we're going to be doing is we're going to be doing a process called fixing. We'll remove carbon dioxide from the air and we fix it to different molecules so we can produce a molecule that's going to be more useful to us, such as sugar. This process will be happening in the stroma, not the stoma, but the stroma of a chloroplast, which is the liquid-filled region outside of these thylakoid discs. So in the light reactions, again, sunlight is used by splitting water as well to produce ATP and NADPH. Oxygen was a waste product of the light reactions. Now we're going to use those two forms of energy to remove carbon dioxide from the air and produce sugars. So our inputs are going to be carbon dioxide itself, which will turn into sugar. All right, either glucose or actually something smaller called G3P. We'll get to that in the next slide. And then the ATP that we use will be oxidized back into ADP and a third phosphate. They'll go back to the thylakoids to get recharged. And we will also then oxidize NADPH back into NADP+, which can also go back to the thylakoids to get recharged during the light reactions. So these two processes are really working together. One can't exist without the other. The Calvin cycle needs energy from the light reactions. The light reactions then need the oxidized forms of those two molecules to continue the process. Alright, so let's look at the steps of the Calvin cycle. What I've done for you is I've broken it down into 4 general steps. All right, I could break them down into a dozen or more if I wanted to, but I think it's a little bit easier just to kind of break it up into 4 chunks. So let's begin, and as we go through the steps, we'll look at it move in action over here in the cycle I've produced. So step 1 is 3 molecules of carbon dioxide, so 3 single molecules of carbon dioxide will combine or be added to 3 molecules of something new that we call Ruby P. Ruby P is a 5-carbon sugar. So once we add a carbon dioxide to each of these 3 Ruby Ps, the first stable intermediate that we're going to make in this cycle, which will be over here in this location, are going to be 6 small 3-carbon sugars called PGA. And PGA stands for phosphoglyceraldehyde, but you'll need to remember that, just PGA for short. So right over here, let's take a look. All right, here are my 3 molecules of carbon dioxide. Any black or grayish dots that you see represent a carbon atom. So we're going to have 3 carbon dioxide atoms coming into the system, which means I'm bringing in 3 carbon atoms. And this is a process called carbon fixation that I had mentioned on the previous slide, where you remove carbon dioxide from the air and you're going to fix it or attach it to another molecule, which is going to be Ruby P. And we're going to put that right here. Alright, so these are our starting molecules for the cycle. Every cycle begins somewhere and has to end at the same spot. So we're going to begin with 3 molecules of Ruby P, and when we're done going through the whole process, we will end with 3 molecules of Ruby P as well. Each Ruby P has 5 carbon atoms, so these black dots here represent carbon atoms. So if I start out with 3 Ruby Ps, each having 5 carbons apiece, you could also think of it as I start this whole cycle with a total of 15 carbons. 3 molecules times 5 carbons apiece. 1 carbon dioxide is then added to each of these 3 Ruby Ps. And our first stable intermediate products we make in this reaction are called PGA. So I have 6 of them here, you can see. Each PGA has 1, 2, 3 black or grayish dots, which represent carbons. So now I have 6 molecules with 3 carbons apiece. 6 times 3 is 18. So I went from 15 total carbons to begin with, and now I'm at 18. And that's because 3 carbon atoms came in the form of 3 CO2s. This whole process of carbon fixation was done by an enzyme called rubisco. All right, there are dozens of enzymes that do this whole process here, but we're only going to be focused on one because it's the one that gets the whole ball rolling. So rubisco over here I have spinning will fix or add a carbon dioxide to each one of these 3 Ruby Ps to get to our first stable intermediate. It's an intermediate because it's not going to be our final product, called PGA. All right, PGA isn't what we want though. We want a different molecule. So in the Calvin cycle, we're going to use some energy, 6 ATPs and 6 NADPHs, to help us convert the 6 PGAs over here into a sugar form that we do want called G3P. G3P stands for glyceraldehyde 3-phosphate. And we're going to have 6 molecules of them. And again, each G3P is a 3-carbon atom. So here's our energy expenditure right here. All right, so we're going to be using the phosphate groups and the electrons and hydrogens from NADPH to reduce these PGA molecules into a different form, and here's our G3Ps. Now, the diagram shows you that they really look the same except for a different name, but our focus isn't the chemical structure right now. It's really just tracing the carbon atoms. So, as I had 6 3-carbon sugars up here for a total of 18 carbons, I now also have 6 3-carbon sugars. Trust me, they do look different molecularly if you could look at them. Again, each G3P has 1, 2, 3 carbons, so 6 times 3 is, again, we have 18 carbons. Now, G3P is the final product that we want to remove from the Calvin cycle, not glucose, actually. So at this point, what I can do is I can remove just one single G3P molecule right here. So if I remove this one right here where my laser pointer is, I'm walking away right now with one single 3-carbon sugar of G3P. That's not glucose. Because if I want this cycle to continue, the remaining 5 G3Ps here, I got to use them. Unfortunately, I can't take them away from the cycle. I have to use them, even though it's what I want, to help regenerate these original 3 Ruby P molecules. So I've taken away one G3P. You can think of this as those original 3 single carbon dioxide molecules that entered the system. Now I'm removing them in a different form, and this form has more potential energy than those 3 CO2s did when we started. Our fourth and final step is now going to be to take those 5 G3Ps and turn them back into the 3 Ruby Ps. To do that, we have to expend a little bit more energy. We expend 3 ATPs. And with those 3 ATPs, as well as additional enzymes that we're not going over, we can cut all these G3Ps apart, use the ATP, and reassemble them back into the original 3 Ruby P molecules that we began with. So again, if you look at these 5 G3Ps and you do the math, I have 5 of them. Each one has 3 carbons. 5 times 3 is 15. So these 15 carbons have to get turned back into the original 15 carbons that we began with, which were 3 Ruby P molecules. So in summary, one turn in the Calvin cycle is going to cost you total 9 ATP, 6 from this step and 3 from here, and 6 NADPH molecules. That helped you reduce PGA into a new form, which is G3P. Don't forget, reduce means to add electrons. We produce, again, one G3P. And that is all we really remove from the system. So when we look at a formula for photosynthesis, we always see 6 CO2 plus 6 water produces C6H12O6, which is glucose, right? as well as 6 O2 or oxygen molecules. Well, we didn't really make that during the Calvin cycle, did we? We made G3P, which actually only has, each G3P has 3 carbons, 6 hydrogens, and 3 oxygens, and a phosphate. So if I want to make glucose, what we do is you would take 2 G3Ps, remove the phosphate group from each, attach the remaining 2 molecules together to get glucose. So that means to make glucose, it's going to take 2 turns, not just 1. 2 turns of the Calvin cycle in the stroma to produce 1 6-carbon sugar of glucose. So that means these numbers up here have to be doubled. So every glucose is going to cost us actually 18 ATP and 12 NADPHs to make 1 6-carbon glucose. I always find the easiest way to remember this is, remember every turn costs you 9 and 6, or 9 ATPs and 6 NADPHs, and every glucose molecule takes 2 turns of the Calvin cycle. So if I remember it that way, instead of 18 and 12 per glucose, let's say I want to make 2 glucose molecules right here instead of 1. Well, 2 glucoses, with each glucose taking 2 turns of the Calvin cycle, that would be what? 4 total turns of the Calvin cycle to make 2 glucoses. So then I would do 4 turns times 9 ATP to give you 36 ATP to make 2 glucoses. Or again, if I want to make 2 glucoses, 4 turns times 6 NADPHs per turn would give me a total of 20 NADPHs to make 2 glucose. So hopefully that'll make it a little bit easier for you to remember it in those terms. Again, lastly, just to look at it in a different fashion here. All this is happening once again in the chloroplast. The light reactions, we're using light and water in the thylakoids to reduce NADP plus and ADP back into ATP and NADPH. These are the reduced forms. Then out here in the stroma, we're going to take carbon dioxide from the atmosphere, fix it to Ruby P, attach it with the help of Rubisco, and we make 6 3-carbon sugars called PGA. And then we're going to use the energy from the light reactions, the ATP and the NADPH, to further reduce PGA, so give it more electrons and hydrogen atoms, and turn it into 6 G3Ps. From there, we remove just 1 3-carbon G3P sugar. The remaining 5 times 3 carbons is 15 total. And a little bit more ATP from the light reactions to regenerate those original 3 Ruby Ps, which each had, again, 1, 2, 3, 4, 5 carbons. 5 carbons times 3 molecules of Ruby B is 15 carbons total, and we are back at it, and we can continue the cycle. So spend the time to memorize these molecules, Ruby P, PGA, G3P, how many carbons they have apiece, and how many molecules of each that we have at any given point of the Calvin cycle.